Okay, guys, so let's recap the main concepts that we are responsible for knowing in chapter one. And that is, remember, we said we're going to be studying a two in one. That anatomy, anatomy, and the physiology. The anatomy and the physiology. And we said that anatomy is another word for structure. So the anatomy is the structure, and the physiology represents the function. So the physiology represents the function, and anatomy represents the structure. And we said that the relationship is interchangeable and interdependent. You can never have a proper physiology unless that you had the proper anatomy that makes it happen, that makes it successful. And you will never have the proper anatomy except that it was to serve the proper physiology and the function. Remember, I gave you an example that if you had a knife and this knife is able to cut, the knife is made of steel, and a hammer is made of steel as well. And the hammer can hammer down the nail, and the knife can cut a tomato. So they have two different functions, one to cut, one to hammer down. But the reason is because the structure is different. One is sharp, one is not that they are all made from steel. You know, people, they're all people, but one of them is a doctor, the other one is a police officer. They're all people, but they have different functions, so they look different. One has a weapon, one has utensils because they're a chef, one has a stethoscope because they are doctors. So your structure is what gives you your function. That's exactly what we're going to be doing in this lecture and in this course. We're going to be making the connection between the structure and the function of all the tissues and the organs. Why does the stomach tissue look like that? Because it needs to secrete the acid to help mechanical digestion and chemical digestion, and the contraction that crush, and the contractions that are the stomach contractions that are responsible for grinding the food and preparing it to be absorbed in the intestine. So you will be making the connection. You will look at the heart cell, you will see it's full of cytoskeletal proteins. We're going to learn the cytoskeletal proteins are the scaffolding proteins. These are the proteins that are responsible for giving the cell structural, mechanical integrity. So you look in the microscope, the cardiac cell has a lot of that, has a lot of cytoskeletal proteins because it needs to pump blood, it needs to pump a lot of blood. Will the neuron, the nerve cell that is responsible for sensory input, processing, integration, and generation of output, will that cell have a lot of cytoskeletal proteins? It will not, because it doesn't have that function. The function is requiring a particular tissue structure to help it happen. So we're going to be looking at the structure of tissues, structure of organs, and the structure of organ systems. And please don't forget this pyramid. Please don't forget this pyramid. This is the pyramid that represents the hierarchy of life. And the hierarchy of life is, we've learned that the cell is the most basic structural level. The cell is, many cells give us tissues, and many tissues give us organs, and many organs give us organ systems, and many organ systems give us organisms, and many organisms give us a population of organisms. So we have learned that from organs and above, that is known as gross anatomy. Gross doesn't mean here disgusting or nasty. Gross means very wide and big, and you can see it with the naked eye. Your microscope can see as small as how much? What's the smallest level you can see with? What is the smallest level that you're able to see with? That's going to be how much? 0.1, that's going to be 1 millimeters. If you are 1 millimeters and more, you will be viewed with the naked eye with no problem. If you are 0.9 millimeters, you cannot be seen with the naked eye. Who do we need so we can see you now? Microscope. Excellent. You're going to need the microscope. So gross anatomy is what you can see and above, which is the stomach, the heart, the intestines, and so on. Now, if you examine here, we've seen that the cell is the most basic unit of life. Remember we said people live in the car, people live in vans, but actually the smallest living arrangement is the apartment. The apartment is the smallest living place that somebody can be in. It has a bathroom, it has a kitchen, it has a dining room, and it has, a bedroom. Then that's the smallest. Then multiple apartments give you a building. Many buildings give you a neighborhood. Then you have a county. Then you have a borough. Then you have a state. Then you have a country. So that's the basic unit. The basic unit of life is the cell. But we realized that there is an invisible, more basic unit below than that, and that is known as the subcellular compartment. That's even smaller than the cell. That's less than the cell. So the subcellular compartment is going to be made of the organelles. Are going to be made of the organelles. These organelles, what are they? The organelles are the organ of the cell. So your organs are your brain, your stomach, your heart, your intestine, but actually the cell, we cannot call the mitochondria the organ of the cell. We call it the organelle. Okay, so we're going to be going through those. Then, because we learned that if something is wrong with the cell, it's not because of the cell itself. If your apartment on fire, it's because the stove was left unattended or the iron was forgotten next to the carpet. So the same thing. If the cell is destroyed, it's because something was wrong inside the cell. Because something's wrong in the organelle. Something is wrong in the nucleus, like cancer. That's a nucleus problem. There's uncontrollable cell division. The cell is constantly dividing and dividing uncontrollably. That's a nucleus problem. Sometimes you can have a problem with inflammation and necrosis, where the plasma membrane now is fractured. So that's a problem of the plasma membrane. The nucleus was fine, but the whole cell is dying because something inside. So we learn now when we want to treat with medications, we target those stuff that are inside the cell, subcellular, below the cell, that are enabling the cell to survive. Then we're going to be discussing even beneath the subcellular compartment, you have the all important subatomic. Excellent, very good. The subatomic compartment. The subatomic compartment. This subatomic compartment is talking about the protons and the neutrons and the electrons. You will learn that the atom has a nucleus. The nucleus of the atom is not the same thing as the nucleus of the cell. Students mix that up all the time. The nucleus of the cell, inside it is the DNA. The nucleus of the atom, inside it is no DNA. Inside it is the proton, the positively charged protons, and the neutral, zero, the neutral neutrons. Anybody heard of neutrons and protons? Those are inside the atom. Then around, who's spinning around? These are the electrons. So those are the subatomic particles. So we're going to need the microscope to view anything beneath the 1 millimeters. And what do we call that now? If above 1 millimeters, you can see it with your eyes, we call that gross anatomy. We call that gross anatomy because it's above 1 millimeter, above the eye. What about below 1, which you need the microscope for? What do we call that one now?
Cytology and histology.
Okay, cytology is the study of the cells. Excellent. And histology is the study of the tissue. Excellent, 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 excellent.
Compass, though. There's a word that means both of them, but I can't remember.
A word that means both of them.
No?
Do you mean histopathology?
No, that's the disease one.
Yeah, very good. The histopathology is more like the disease. But no, cytology is studying the cell when you're focused on studying the cell, and histology when you're focused on studying the tissue. Okay, so now we describe what is anatomy and what is physiology. Gross anatomy, we discussed that, didn't we? We discussed what is gross anatomy. We have discussed what is gross anatomy, and we have discussed what is histology. Is that clear? We have discussed histopathology, cytology. Excellent job. And we have learned what is ultrastructure, and that is when we learn, ultrastructure is when we learn up to the molecule. The molecule. Robert Hooke is a contributor to the development of the microscope. The microscope we have today. Today we have some very interesting types of microscope. There's the electron microscope that sees very small particles. We have the compound microscope. That was the early one that enabled us to finally see that we are only the tip of the iceberg. Gross anatomy is like 10%. The histology and the cytology is like the 99%. So Robert Hooke is one of the contributors. He made the lenses. Then after Robert Hooke, Andrew Van Leeuwenhoek came through, and he was able to develop the first most efficient microscope based on the work that was done before him by who? Who was the scientist before Andrew Van Leeuwenhoek? That was Robert Hooke. That was Robert Hooke. And then Andrew Van Leeuwenhoek was a haberdasher. Anybody heard of a haberdasher? A haberdasher is somebody who's responsible for, the linen and the fabric and that type of person. So he needed like a lens to see the details of the fabric. Then while doing that, he was able to see microorganisms. That's how the magnification was. We need to define the difference between magnification and resolution. Magnification is making things wide, making things very wide, and you can see them. Something is like 0.5 millimeters. That cannot be seen. So you magnify it, you increase the magnification by 10 times. So now 0.5 millimeters, that's going to be 5 millimeters now. So using a lens. So you will learn about the magnification. Resolution does not make things wider. It does not blast things open. Resolution enables you to see the fine details. Enables you to see the fine details. So you need to understand the difference between the resolution and the magnification. We also must understand the cell theory. What is the cell theory? The cell theory says that all living things are alive because they are made of cells. You are not alive except that you are made of cells. The number one requirement for you to be called alive is for you to be made of cells. If you're not made up of cells, you're not alive. Number two, your cells have to be living cells because the cadaver is made up of many cells, but they are dead cells. This chair is not made up of cells, so it's an inanimate object. It doesn't have cells, so it's not living. Number two, all cells come from other cells. Cells don't come out of nowhere. I'm here because my father was there. My father is there because my grandfather is there, and so on. So cells must come from pre-existing cells. formerly pre-existing cells. Is that clear? Now we're going to learn about the scientific method. This is something that we learn in biology. Like you said, your lab teacher is a biologist. So we begin biology by first understanding inductive versus deductive reasoning. Raise your hand if you ever heard of the difference between inductive and deductive reasoning. What's the difference between inductive and deductive reasoning? What's the difference between inductive and deductive reasoning? Okay.
Inductive reasoning, you basically make a hypothesis, but you deduce what can happen or cannot happen.
Okay, so deductive reasoning, you begin from very big and you narrow down to the deductive reasoning. And building up is, that is inductive reasoning. So now let's explore that some more. What do I mean by deductive reasoning? Deductive reasoning is when you have a pre-established premise or a pre-established fact. You have a pre-established premise and you have a pre-established fact. For example, when you say all mammals, what do you know about mammals, guys? What's a mammal?
It's a living creature that requires a male and a female in order to have offspring.
It breastfeeds its baby because a cockroach also has male and female.
But I don't think they're required for each other, right?
Yeah, they're required, but they don't breast milk.
Animals, not insects.
Yeah, animals. So the mammals breastfeed their baby. That's a mammal. Another thing about mammals is mammals have hair. Cockroaches have hair. So reptiles have hair. So mammals have hair. So now you know one thing. All mammals have hair. True or false?
True.
True. All mammals breastfeed their baby. True or false?
True.
So that's a pre-established premise. We know that. That's a pre-established fact. We already know that. Let's say now I tell you the whale is a mammal. Then you say, oh, the whale is a mammal, then it must breastfeed its baby, then it must have hair. That's deductive reasoning. You use deductive reasoning unknowingly because you deduced what the whale is all about based on your former knowledge of what a mammal is. Is that clear? So deductive reasoning is when you have a known fact and then you base upon it. Inductive reasoning doesn't work that way. Inductive reasoning is when you do a specific experiment or trial or diagnostic. When you do a specific diagnostic or a trial or experiment in order to find out something. For example, you take the thermometer, you measure the temperature, and you see that the temperature is about 108 degrees Celsius. So you think that this person has a fever. That's inductive reasoning because you made your reasoning based on an experiment, a trial, a diagnostic. Is that clear? If I tell you I measure the blood pressure and the blood pressure is 160/90, that's an example of 160/90, that's hypertension. So that's an example of inductive reasoning. I would have never known that this is hypertension if it wasn't for that sphygmomanometer that showed me that the blood pressure is high. So I didn't rely on a pre-established premise. I investigated using a diagnostic, using a particular modality to help me discover the problem. So that's not deductive, that's inductive reasoning. Is that clear?
Yeah.
Okay, so in order for us to be able to investigate, we have a stepwise process known as the scientific method, known as the scientific method. The scientific method is multiple consecutive steps, 1, 2, 3, and so on. Number one, you have an observation. This observation is based on what? On your senses. You can never observe without your senses. You have to see something. You have to hear something. If you do not see, you do not hear, you cannot make an observation, can you? If Isaac Newton did not see the apple fall drop on the ground, would he have ever had an observation and then investigated gravity? So all observation is based on sensation. So the people who are sensitive are very brilliant because they're always observing something. But people who are, their senses, you see the very elderly people, their vision is not good, their hearing is not good, they get robbed all the time, they get stolen, why? Because they are not observing things properly, because their senses are deteriorating. Does that make sense? And the babies always get the baby abuse, they always go through a problem. Why? Because they are still developing and their senses are not well nourished for them to make observation. So the most important thing is the observation. And the observation will not come except that we had a hypothesis. Then the observation, if you had an observation, you have to make a hypothesis. And the hypothesis definitely has to be a testable hypothesis. A testable hypothesis. Hypothesis that I can test. You cannot say salt water freezes after fresh water. If you have a pure water cup and a salt water cup from the ocean, you put them on a freezer, which one is freezing first?
Fresh water.
The fresh water. So then that's an observation. You observed. You saw which one froze first. That's not a hypothesis. Was that a hypothesis? If you look at which one freeze first and you find out, is that a hypothesis?
Observation.
But if you explain the observation, if you take a step further and you say, I believe that the fresh water froze before the salt water because the salt water lowers the freezing point, then that's a testable hypothesis. I can test that. So I can put four different cups. On the first one I put 5 milligrams of salt. The second one 4 milligrams of salt. Third one 3 milligrams of salt. The fifth one 2 milligrams of salt. The last one 1 milligram of salt, and the one is empty, no salt in it. That's a control group. Then I put them all in the freezer. If I see that they start to freeze up in an ascending order from the least salt to the most salt, then the salt was my determining factor. Is that clear? So my hypothesis has to be testable. What if I say the fresh water freezes first because it's blessed by God? Maybe that's true, but I cannot test that. So that's not scientific. So just because something is not scientific, it doesn't mean it's not true. It doesn't exist. If something is not scientific, it does not mean it doesn't exist or is not true. It just means that it cannot be tested scientifically by scientific means. Then the testable hypothesis, you form an experimental design. And the experimental design, you have independent versus dependent variables. Independent versus dependent variables. Are we clear? Any questions? then we investigate the independent versus dependent variable, then we publish our results, and then we share our results with peer review, with peer review community. These are other experts, other scientists. We don't share with the people on the Facebook, with the people we don't know. We share it on people who are qualified, people who are on our field to give us constructive feedback that helps us develop the experiment. Are we clear? Okay, so now let's go back to the slides. The scientific method is the process of performing science, including careful observation, logical thinking, and proper analysis. Science and scientific methods set standards for truth. We discussed the inductive method and the deductive method. And falsifiability means that if we claim something is scientifically true, we must be able to specify what evidence it would take to prove it wrong. So our experimental design, we have a sample size. Let's say I have 100 patients that have high blood pressure. I want to know, I have a medication. I want to know if it's going to lower blood pressure or not. Am I going to give the medication to all the 100 patients?
No.
I'm going to split them in half. 50 are going to get the actual medication, and 50 are going to get just a capsule full of water. But they don't know it's a medication. To see and reference, my control group is my reference group that I use to find out. My reference group that I use to find out if the variable that I'm testing, to find out if the variable that I am testing is successfully, operating, is the one behind the actual results. So there is bias that must be removed. First of all, the bias of the actual subjects. They know they are in an experiment. They're going to be getting blood pressure medication, so they're going to be expecting their blood pressure to go down. Some of them may say, I'm worried that my blood pressure now, it may not go down even though there is a blood pressure medication, so they're going to get worried. And then, it may go up or may go down. So we don't tell them what the dosage is. We don't tell them which group they're in. Are they control group or experimental group? So that's called single blind. Single blind is when the subjects don't know if they are going to be getting the medication to lower blood pressure or not. And then the control group is the one that is getting the placebo. The placebo is everything except the variable that I'm testing. So they're going to get water, sugar, but they're not going to be getting a medication that's going to lower the blood pressure down. Are we clear? Peer review. Peer review is a critical evaluation by other experts in the field. So that is done prior to funding or publication. It is done by using verification and repeatability of the results. It ensures honesty, objectivity, and quality in the science. Okay, the laws and the theories. So laws are generalizations about the way the matter and energy behave. Please remember the difference between single blind and double blind. What's the difference between single blind and double blind?
Single is the patients that don't know, and double blind is the people that are administrating the medication don't know either.
Yeah, both of them don't know.
Both of them don't know.
Double, yeah, very good. Is that what you wanted to say? Excellent, that's very good. So, law of the nature is a generalization about the way the matter and energy behave, while theory is an explanatory statement or set of statements derived from the facts, the laws, and confirmed hypotheses. The theory of natural selection states that the mechanism by which evolution is going to propagate is called natural selection. And it describes for us how species originate and change throughout the time. They help understand. They help to understand the body. So Charles Darwin is the biologist who first investigated this. And in evolution, you see that there is a change in genetic composition of the population of the organisms. So, for example, when it comes to bacteria, if you take an antibiotic, how much of the bacteria will die? 100% of the bacteria will die? Of course not. But the vast majority, like 99%, the remaining 1% who did not die, due to a mutation perhaps, that 1%, is it going to stay 1% forever? It's going to multiply. It's going to multiply exponentially. The bacteria is going to exponentially multiply. When the bacteria multiplies exponentially, the next time when you give it that medication, will it work? It survived it. So now you're dealing with something that is a survivor. So us human beings, we are the progeny, the offspring of the survivors. Otherwise we would have been dead. You know, this earth has been through many plate tectonics, has been through ice age, so all human beings died except 1% who survived. They kept on and on and on. Then we came out of them. So we are the survivors. So evolution means that the small population who is going to get away from the complete, total, generalized destruction is going to adapt to all the challenges that are around. Adaptation are inherited features of the anatomy and the physiology that evolve in response to the pressures and that enable the organism to succeed. So some human anatomical features is that they can be traced back to the ancestral primates. So early primates were known as arboreal. You must understand this word. What does arboreal mean? Arboreal means climbing trees, dwelling on, they live on the tree like the monkey. And we became able to walk on 2 feet. This is known as bipedalism. And this shows human adaptations. We have some capability of using the prehensile thumb, like using the chopsticks. The monkey has some shared characteristics. People think evolution means that we came from monkeys, that our great-grandfather is a monkey. That's not true. You're not going to look in a photo album and you say to your mom, who's that? She said, that's your daddy. Who's that? That's your granddad. Who's that? That's your great-granddad. Who's that monkey? That's our great-great. That's not how evolution works. Evolution says that we have shared similar biological characteristics with other primates, but it doesn't mean we came from them. Bipedalism, standing up and walking on 2 feet. That's what we discussed in the lab. That standing up, you have superior versus inferior, you have anterior versus posterior, and so on. What's the scientific name for the human being? You and I. Homo sapien. Very good. Homo sapien. Very good. Now we're talking business. This is the most important thing that we need to focus completely on. And this is the hierarchy of complexity. We discussed that. Remember, we said from tissues and above. So we know that molecules are made up of atoms. So if you have atoms, will give you molecules. Molecules will give you organelles. Organelles will give you cells. Cells will give you tissues. Tissues will give you organs. Organs will give you organ systems. Organ systems will give you organisms. And organisms will give you a population. A population. Is that clear? This figure is one of the questions that are on that I put for you. Is the lab section still up there? the lab section that I assigned for you?
Yeah, it was in one of the questions.
Yeah, so please do those.
I haven't.
I don't see anything, and the one I see is locked.
Yeah, because there was one due on week 1. There is another one that's due right before the supposed the lab practical. But now see with your teacher how the lab practical is going to be. But either way, I believe if you have access till October, please, those will help you. Because that question, for example, is going to remove atom, molecule, macromolecule, organelle, cell, tissue, organ system, organism, and it's going to point to a letter A. Tell you what is that? You say that's an organism. What is that? That's an organ system. What is that? Organ, tissue, made up of cells, and so on. So that shows you the hierarchy of life. We discussed that. This is the cellular compartment, the cell. That's the organelle, the subcellular, below the cell. Then you have the molecule, the ultrastructure. This is the atom, the subatomic structure. Are we clear? Yes.
I got a curious question.
Yeah.
So, on the work, I don't know if you saw it, but both homeworks are still the same. If you technically think about it, so I know that one was like 30 questions. When you do week 2, it makes you redo the same 30 questions, like word question for question. I don't know why. Was that
For the, you mean the lab course or the lecture?
For lab.
lab? So there's the first one, which is that one, and then the second one is like 100, like a lot of questions.
Yeah.
But the first 30 of those questions are literally the same.
Exactly. That's what I had in mind. I wanted to give you all the figures that you're responsible for knowing in the lab practical. And every week I will chop up some corresponding to what we cover every week. Some students, they finish all of the 200 like on the first day, and they just keep repeating, reviewing to the day of the exam.
Okay.
Okay? But they will help you if you go over them. But we're gonna sort this out. How if McGraw-Hill Connect, if that's gonna be used. But otherwise, find out. Contact the lab professor about how the lab practical is gonna be. You need to prepare for that. How she's going to prepare for this. So we said that the organism is a single complete individual, and the organ system represents a group of organs with a unique collective function. For example, circulation, respiration, digestion. That's the definition of the organ system. And you have an organ which is representing a structure that's composed of 2 or more tissue types that work together to carry out a particular function. Then we have the tissue, followed by the cell, and lastly we have the smallest particles, that if you break the atom to a smaller particle, what are you going to get? Useless. The most smallest useful substance is the atom. If you break the atom in half, you get nothing. So the atom is the smallest useful with a unique chemical identity. What is reductionism versus holism? Reductionism meaning we study from the top to the bottom. We study the organism, we reduce the organism into organ systems, we reduce organ system to organs, we reduce organs to tissues, we reduce tissues to cells, we reduce cells to organelles, we reduce organelles to macromolecules, we reduce macromolecules to molecules, we reduce molecules to atoms, Holism is the opposite. Holism is when you build up, is when you start. So reductionism is when you go from top to bottom. You reduce all of these structures. But holism is when you build up. You start studying in the other direction. Instead of breaking down an organism into its smallest compartments, now you build up and you join all the compartments to make your big organism. So these are 2 different conventions of studying, the reductionism and the holism. Okay guys, we now need to discuss the difference between anatomical variation and physiological variation. What is anatomical variation and physiological variation? Anatomical variation means that anatomically, structurally, You are different from other people, even though you're not supposed to. So, for example, the anatomy and physiology we're going to be studying in this course, it's for about 80 to 90% of the people. So all the people are going to have 5 fingers. Does that mean you will never see somebody with 6 fingers? That's an example of a anatomical variation. That's a diversity. Not an anatomical diversity, it's an anatomical variation. I had a student that showed me she had a bicuspid aortic valve. So the aortic valve is usually tricuspid. It has 3 cusps. All the valves in your body are tricuspid, except the left atrioventricular valve, known as the mitral valve. We're going to learn that in when we do the heart. What happened with, yes, what happened with her, she ended up with a bicuspid aortic, even though it was supposed to be what? Tricuspid. Did that affect her life? It didn't. It's just like having an extra finger. So you have to understand that even though we study anatomy and physiology, the anatomy and physiology that we are studying now is good enough for how many? It's good enough for only 80 to 90. If you see the 10%, the other 10%, are you supposed to run out the window? So that's normal. That's an anatomical variation. What's an example of a physiological variation?
Would it be plants?
What, how the body would move?
I'm sorry?
How the structure of the body would function to do something?
That's physiology.
Oh, I think you said physiology.
Maybe if you're like blind,
I'm sorry?
if you're blind, you still.
If you're blind, that's a disease, that's a pathology. That's a sensory deficit. You have to understand that physiological variation, meaning we are the same, but I do my functions differently. Like male,
That's what I said.
really?
Yeah.
I'm sorry, the way you articulated it, maybe I didn't understand it. So that's like when you compare a male and a female. Both are is a 22-year-old man and a 22-year-old woman weigh the same? Do they eat the same amount of food? Do they burn the same amount of calories every day? Of course not. That's a physiological variation. The woman has more lipids in her body than her counterpart who's our same age and happens to be a male, because women have more lipids. So that's the anatomical or physiological variation. Having more lipid, more fat in the body between men and women, is that an example of physiological or anatomical variation? That's an example of, is fat physiological or anatomical? That's anatomical, the fat in the belly, in the muscles. So that's part of the structure of the body. Fat is connective tissue, part of the structure of the body. So that shows you a anatomical variation. Physiological variation is how many calories I burn per day. My function is different from you. My function, for example, men used to run out into the desert or into the woods and hunt and get the deer and bring it home, and the woman stay home with the babies. So the man is running and fighting the beast and trying to get the food, and the woman, for example, at the time used to stay with the baby and breastfeed the baby. So she doesn't burn as many calories. True or false?
True.
So that shows you the function is different. The function is different even though we are the same age. So now we're going to look at, when you look at the body, everybody is going to have a particular kidney structure that is familiar to all of us. We all know the kidneys. How do they look? Kidneys look like that, except that the kidney on the right side is a little bit depressed compared to the kidney on the left side. True or false? The kidney on the right side is a little bit more depressed than the kidney on the left side. True or false?
True.
True, because the lobe of the liver, the liver pushes it down. Ever ran on a treadmill and felt like this side is burning while you're running? That is because the liver is squeezing and bursting a lot of glucose to sustain your energy metabolic demand, to provide you with glucose that you can burn and make ATP out of. Any questions?
So when you say right, you're really meaning what we're looking at from our left?
No, remember we discussed the anatomical position. We said it's not my right, it's not your right. It's the right of the anatomical position. And we said, how does the anatomical position look like?
Palms out.
So both palms have to be facing outward, and the face is facing into the front. Both feet, not like this, but they're supposed to be facing to the front. And also, you're not standing on your tiptoes. So when you stand, you can stand anyway, but you have to visualize the subject in the anatomical position. Imagine the doctor says, this Professor Kadir has a right-sided mass. And I go to a doctor in Japan. Is the doctor gonna say, wait, was it your right or my right?
No.
Is the doctor in Mexico gonna say, wait, which right was it? So we needed to make a frame of reference. We needed to make a frame of reference that is uniform to all of us. A frame of reference that all of us got to know. A frame of reference that we're all familiar with. Are we clear?
Now, do they all function the same, even though?
Yeah, yes, they all function the same. They said, remember, anatomical variation in anatomy. They never said variation in physiology. It's a good question. Also the heart. You see this is the aortic arch, and you see all of these branches emanating and coming out of the aorta. Are we clear? So now we need to understand what life is. You know, scientists had a difficult time explaining or defining what life is. What is life? So they still cannot agree to what life is. But they said, we'll do this. Let's write all of our scientific observation of living things. So they realized that all living things are very organized. All living things are very organized. They're organized structurally and organized functionally compared to non-living things. They also realized that living things have cells, because the cell is the basic unit of life. You're not alive except that you're made of what? Cells. And cells only come from pre-existing cells. Very good. They realized that also living things are able to successfully carry out what? Successfully able to carry out. They realized that cells are successfully able to carry out metabolism. Cells are able to carry out metabolism. And cells are, their nervous system is intact, and they are very responsive. They are very, very responsive. And they have the ability to sense and react to changes in the environment. Okay. Also, living things are able to move. Right? Cells are able to move. It's called taxis, chemotaxis. The bacteria is able to move. If you stay in one place, you're dead. So you have to be able to move. You have to be able to move continuously to be called alive. So you see, we cannot define what life is. But we say that if you are alive, you have to have all these things happening. You have to be organized. You have to be made of cells. You have to be able to carry out metabolism. You have to respond to stimuli, and you have to be responsible for movement. Now, if you look at the characteristics of life, you see that there's also homeostasis, which means the ability to restore. Homeostasis represents the ability to restore any deviation. Meaning, if you're too cold, you shiver to become warm. If you're too hot, you sweat to become cool, to cool off. So, sweating and shivering, they both did something in order to restore your proper temperature. If it wasn't for the sweating, you're going to be very hot. You're going to end up with a hemorrhage. The runaway fever, you're going to end up with a form of a seizure. In addition to that, what happens if you're too cold and you're freezing? You're going to keep on colder and colder and colder. You're going to end up with a coma. That's why your body is going to do shivering response. Shivering thermogenesis. Shivering thermogenesis. That enables you to survive the cold. Evolution represents the genetic change from generation to generation that occurs due to the mutations. So now we talk about the physiological variations. Physiological variations are variations in sex, age, diet, weight, physical activity, genetics, and the environment also. Are we clear? So that means when we look at a reference man and a reference woman, the reference man weighs about 144 pounds, while a woman is also 22-year-old pounds, but she's 22 years old, not 22 years pounds, 22 years old. But how many pounds does she weigh? 128 compared to the man. So she burns $2,000. She can burn $2,000 a day actually. But she burned 2,000 kilocalories. It happened to me. You know, a woman can burn $2,000 a day. But anyways, the reference woman would burn 2,000 kilocalories a day, and the man would burn 2,800 kilocalories a day, because the man is physiologically and anatomically supposed to be the hunter-gatherer, supposed to be going out doing work, not like many of, unfortunately, the men nowadays, they're sleeping all the time. But they're supposed to go out and work and lift things and work physically and mentally, you know. Yeah. We're supposed to do that. Now we want to talk about the very interesting concept of homeostasis. What is a homeostasis? Homeostasis is the ability to remain, at a safe physiological range, no matter what's happening on the outside environment. So let me explain that some more. This is actually very, very important. I hope this thing is working. Okay, so let's discuss the difference between hyper and hypo stimulation. So now we have, for example, if we look now, we have If we now look, we see that there's always a sensor. There's always supposed to be a sensor. A sensor is gonna alert that there is a deviation. So usually we don't want hyper something, we don't want hypo something, we want right the balance, the middle range, which is U something. Hyper something is plus, plus, plus. Hypo something is minus, minus, minus. Plus, 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 minus, minus, minus. That's very bad. When you're 2 plus, you know, it's not the more the better. So what is so much plus plus? That's an example of, for example, if we look at hypertension, if we examine hypertension, that means the blood pressure is too high now. What's the opposite of hypertension? Hypotension. That's when the blood pressure is too low. Which one is better?
Neither.
They're both bad. Very good. When it comes to sugar, you have hyperglycemia, hypoglycemia. We don't want hyperglycemia or hypoglycemia. We want euglycemia. You have to say the whole thing. Euglycemia, eutension. When it comes to the temperature, we don't want hyperthermia, it's too hot. We don't want hypothermia, it's too cold. We want euthermia. We want euthermia. So U represents homeostasis. Students always, wonder what's the difference between the what does homeostasis mean? Some students tell me that homeostasis means balance. That's not true. Homeostasis means you remain constant no matter what's happening on the outside. The outside is snowing, you shiver, you will cool, you will warm up. So for example, if they put the thermometer, you're at 37 degrees Celsius. You step outside, it's cold, with the thermometer still in your mouth. Your body's gonna shiver, your temperature is still 37 degrees Celsius. If it's too hot, you will see that you're gonna sweat and your temperature is still 37 degrees Celsius. So there are mechanisms to fight hyperthermia, hypothermia, and we will end up with euthermia. Euthermia. Are we clear? So restoring U, that's homeostasis. So how does that, now we're going to talk about the steps, the successive steps that lead us to restore homeostasis. What are the steps? Step number 1, a sensor. What's the sensor? A sensor is a receptor. A sensor is a form of receptor. A sensor is a form of a receptor. Is that clear, guys? Any questions? A sensor is a receptor. How many kinds of receptors do we have? We have photoreceptors that detect light. We have photoreceptors. We have chemoreceptors. We have mechanoreceptors. We have osmoreceptors. We have also chemo, mechano, osmo, photo. What else do we have? Thermoreceptors. So the thermoreceptors are the sensors for heat and cold. Osmoreceptors are sensors for the osmotic balance. The homeostatic osmotic balance is 300 milliosmoles. Mechanoreceptors, they detect any mechanical stimulation. Like in the blood vessel, when the blood pressure is running so hard in the blood vessels, striking the blood vessels end up dilating. The blood vessels dilate severely, and that is gonna now is gonna be sensed. You know, like when you're sitting and somebody running on the roof, it's because they slamming on the ground, and you can hear that. So what happens is how does your brain know your blood pressure is too high? Because those mechanoreceptors are sitting around the blood vessel. When the blood pressure is so high, the blood is gushing and rushing and hitting the walls of the blood vessel, and the blood vessels are dilating. And when they dilate, who's going to be stretched out? The mechanoreceptors. So our sensors, we have different types of sensors. The sensors are going to sense an input. They're going to sense an input. A input is always sensory because it's coming from an input is always sensory because it's coming from the receptor. The input is always sensory and it's coming from the receptor. And the input is going to go to a control center. The input is going to go where? To a control center. To a control center. The control center is called a control center. It has another name. A control center, integration center, control center, integration center, and that's going to generate an output. That is going to generate an output. That is motor. Is going to generate a motor output. An input that's sensory, and an output that's motor. Input that's sensory, and an output that's motor. Then the output is going to go to who? It's going to go to the effector. The effector is either a gland or a organ, or an organ. A gland or an organ. This is really, really, really important. Step number one is a sensor who's going to detect there's some hyper problem or hypo problem, hypertension, hypotension, and then there's going to be a sensory input. That's step number two. And then the control center is going to be step number three. And then the output is going to be step number four. And lastly, the effector is step number 5. Are we clear? Are we clear?
Yes.
So now let's look at these different responses. Let us begin with the temperature. Let us begin with the temperature. If the temperature went up, is that hyper U or hypo? Hyper what? Hyperthermia. You guys are very good. That's hyperthermia. Who's going to detect the sensor who will detect the hyperthermia? Is it a photoreceptor, chemoreceptor, mechanoreceptor, osmoreceptor, or thermoreceptor? Excellent. That's a thermoreceptor. Thermoreceptor is going to send the input signal to who? To the nucleus, to the thermal nucleus in the brain. It's going to tell the thermal nucleus in the brain is too hot or too cold. Somebody said too cold?
No, it's too hot.
Too hot. So it's going to say, the input is going to say, it's too hot, it's hyper. What do you think the control center is going to tell the effector? It's going to tell it, become more hot or become cold. So the output
motor signal is going to become cold or hot?
Cold.
Cold. Now look at this. That's how you're going to be able to tell negative feedback. Negative feedback is when you have a reversal. You have an input and an output that are the opposite. If you're driving your car going at 80 miles an hour, then you hear the police siren go off because you cross the speed limit. Then you press on the gas and you lower your speed. You used to go fast, now you lower the speed. Is that positive feedback or negative feedback?
Negative.
That's negative feedback. When before and after are the opposite, that's negative feedback. When you correct a problem, when you fix it, because you were going too fast, now you fixed it, that's negative feedback. People think positive feedback means positive is good. No. Negative feedback is good because it's the opposite. What about if you're going so slow? The 18-wheeler gonna run on top of you. So now you press the gas. You were going too slow, now you're going so fast. Is that negative feedback or positive feedback?
Negative.
That's negative feedback because you used to go slow, now you go high. You fixed it. But imagine you going slow and the 18-wheeler is behind you and you press the brake now even more. What is that now? That's positive feedback. Or you go too fast and your speed goes 80 miles an hour and the speed limit is 65, then the police are running after you to stop. You're like, they already have a warrant on me. So you just press the gas and you go 90. That's negative feedback or positive feedback? That he thinks he's doing negative feedback in his mind. He thinks he's doing negative feedback. He is not doing negative feedback. He's doing positive feedback. So when you don't repair the problem, that's positive. Negative feedback when you fix it. So now, we need to talk about who's the effector. You know when you call 911, let's say I see a fire in a house. You see somebody being assaulted. Somebody sees somebody in a coma. So you need an ambulance. I need the police officer, and she needs a fire truck. Are you gonna call 911 and I call 922 and they call 933, or we all gonna call the same 911? So the same thing, we all call the brain. If it's too hot, we call the brain. If sugar is too much, we call the brain. If it's too low, calcium is too low, we call the brain. But the brain is very good at responding properly. So the 911 is gonna send me the police. If I need the police, they're not gonna send me the ambulance. If you need the ambulance, they're gonna send you the ambulance. They're not gonna send you the fire truck or the police. So the same thing. But you need to know, the one who's gonna come, the police when they come, the fire truck when they come, the ambulance when they come, that's the effector.
So
Yeah.
calling 911 would be the receptor?
So you're the receptor who called, yeah, because you gave the input. 911 would have never known that there's a thief.
So what would 911 be?
911 would be the integrating control center.
Okay.
They are the one who will integrate, who will receive the input. So look at the map, this messy map. You will see that the control center is in the middle between the input reception. The receptor is the one who's gonna send the input. The integrating center is gonna process it. The output signal is gonna send it out.
So the output is 911 sending out, and then the effector is when they get there.
The effector is the one who's gonna do it. The effector is the actual fire truck is the effector, if you needed it. The police is the effector, and the ambulance is the effector. But we're not doing 911 here, we're doing the body.
Yeah.
So when you're too hot and your sweat glands secrete sweat because it's too hot, who's the effector now? The sweat gland. Because they secreted the sweat. Who told them to secrete the sweat because it's too hot?
Is it the brain?
The brain, the integrating center.
The brain?
Hmm?
Is it always the brain?
No, not always. Sometimes you see the pancreas. The pancreas is the one who's gonna taste the blood. The blood, too much sugar. Let me secrete insulin. Too little sugar. Let me secrete glucagon to put it on the right ratio. You really need to know that. You really need to be very good at who's the receptor, who's the input signal, who's the processing control center, who's the output, who's the effector. So I told you the effector is always a gland or an organ. For example, when it's too hot, let's continue with hot. Hyperthermia is thermoreceptor, input signal, sensory signal, then a control center, that's the integration center, then the output, motor stimulation is going to cause an effector who's gonna cool you because it was hyperthermia. Who's gonna cool you now? Who's gonna cool you when it's too hot? Who's gonna cool you when it's too hot? The sweat gland. That's the effector. And is the sweat gonna go endlessly on and on and on, or is it gonna stop at some point? It's gonna stop when? When you return to the homeostatic temperature. Now let's do the opposite when it's too cold. When it's too cold, that's hypothermia. Hypothermia. Hypothermia gonna send thermoreceptors to the brain. The brain is gonna send out output to who? The muscle, to make the muscle shiver. When the muscle shiver, who's the effector now? The muscle is the one who shivered to cool you off. Is it going to shiver forever or just to return you to normal? Return you to normal. Are we clear?
Yeah.
Any questions? You will see also when it's too hot and cold, it's not just the sweat gland, it's not just the muscle. 1 second, I will get to you. And the muscle, also the blood vessels. Have you ever worn a ring in the summer and it felt too tight?
Yes.
In the summer, in addition to the sweat, your blood vessels, always when it's too hot, they vasodilate. They are engorged, really full of blood because they are shunting the heat in your core out to the periphery. Is that clear? Out to the periphery. So they vasodilate. But when you're too cold, are they going to vasodilate? Because if they do, they're going to be full of hot blood. Blood is going to escape. But if you keep them in check, if they make them constrict, the blood is not going to be leaking to the outside. Because if it goes to the outside of the skin, it's going to flow to the air. You're already cold. You have a little bit of heat. You want to conserve it. So you do vasoconstriction. So who's the effector here? The blood vessels. The blood vessels. Are we clear? Yes.
So, for example, with the sweat glands, right? After you sweat and you want to stop sweating, does the receptor need to send another signal to stop sweating?
Exactly. The receptor is, excellent question. The receptor is the one who is monitoring. We're too hot. How are we now? We're secreting sweat. We're cooling off. We're cooling up. Oops, that's it. We have reached 37 degrees Celsius. We need to stop now. So the receptor is the one who shut it down. Excellent question. Other questions? Any questions? Good God. This you understand. Negative feedback and positive feedback, you're good to go. Yeah, hello. Okay, we're almost there. We're almost there. We're almost there. We're almost there. Okay. So this homeostasis, the ability to detect change, receptor, detect change, activate mechanism to opposite integration center, and maintain relative internal condition. So we have so there we go. Please look at these two examples. We talked about those. If it's too warm, the skin blood vessels dilate and the sweating begins. That's a heat losing mechanism. Because when the blood is too hot, we send it to the skin around the surface so the heat can go out to the window, can go out to the skin. If it's too cold, the blood vessels constrict and the shivering begins. So if you look here in your room, please look here. The room temperature falls. That's the key word. Falls. Falls. What is that? Hypothermia. Excellent. The thermostat activates the furnace. What's the thermostat? Activates the furnace. Activates the furnace.
That's the input.
Activates the furnace. Please think.
Oh, that's the output.
The output is the arrow, but the thermostat is who?
The control center.
Excellent. Because the thermostat is the one who activated the furnace. Who is the furnace?
The furnace is the effector.
Effector. Perfect. You got it. Because the thermostat can tell the effector to start. If the effector does not start, we're gonna be cold. It's the one who did the effect of restoring the temperature is the furnace. That's the effector. But the furnace wouldn't have known to start unless it got the message from the call center, from 911, from the thermostat. That's the integrating center. So sometimes you can have a two-in-one. The thermostat is actually a receptor as well as integration center. Like your pancreas. Your pancreas tasting the blood, too much sugar, and is an integration center, secret insulin. So you can have two-in-one. But it doesn't matter, you have two-in-one, you have three-in-one. In the end, there's an order. There's always sensor number one. You cannot have a loop if you don't sense anything. You sense, you observe a problem, then you send the input, then there's gotta be a particular type of a control center, then you send out effector, and then you see the heat output. The heat output is generated by the furnace, and then the room temperature rises. You see below we had falls, now we have rises. Is this positive or negative feedback? Good, you really got it now. And then the thermostat shut off the furnace because we returned to normal.
Homeostasis.
Homeostasis has been achieved. It may we just keep going the furnace on and on. We're gonna melt now. So we gotta stop. We gotta know when to stop. Students think that the loop ends at effector. No, the loop ends at stopping.
At homeostasis.
At negative feedback to stop.
The first source of the loop would be number 6.
The first source of the loop would be no. Room cools down. We have achieved homeostasis. That's the end. Because now we have cool down.
So thermostat shuts off furnace would be what exactly?
So thermostat shuts off the furnace. Yeah, that's the same thing as cool down because you shut off the furnace to cool down. So that's going to be homeostasis because we negative feedback. We shut down because you went to the speed limit. You were going too fast. You press the brake to reach the speed limit. You stop pressing the brake anymore because you have reached that homeostatic speed.
Okay.
Okay? You gotta see more examples. Oh, you gotta see this all semester long. You gotta see different systems restoring temperature. There we go. What's our set temperature? 37 degrees Celsius. That's our set point. If we go up to 37.5, what is that now?
Hypertension.
That
I mean, hypertension.
hyperthermia.
Yeah.
When it's too hot now, what are the two responses? Let's begin with the first response. What's the first response when it's too hot? The sweating. You sweat to cool. Is that the only thing you do when it's too hot?